Welcome to the deep dive. So, our mission today is really to dig into what's next in cancer therapy. We're looking beyond CRT cells.
Yeah, which have been amazing, but limited.
Exactly, revolutionary for sure, but they hit roadblocks. We want to explore what's being done to find safer, more effective ways, particularly for, solid tumors.
That really is the core challenge, isn't it? I mean,
Yeah.
CAR T-cells, these chimeric antigen receptor T-cells, they absolutely changed the game in oncology. We saw curative outcomes in liquid tumors like B-cell malignancies,
Right.
and they're even being used now in some autoimmune conditions. But like you said, there's this huge wall when you get to solid tumors, which are, well, most human cancers.
Okay, so let's quickly lay out those main roadblocks. The big three for CAR-T. First up, toxicity.
Oh, yeah.
We're talking serious, sometimes life-threatening things. Cytokine release syndrome, CRS neurotoxicity, even graft versus host disease if you're using donor cells. It's intense. Needs ICU-level pair sometimes.
It really does. The T-cell response is so aggressive, it can cause this systemic inflammatory storm. It could be catastrophic.
Okay, challenge number two, solid tumors. You mentioned the wall. Why are CAR-Ts just not working well there?
Well, fundamentally, they struggle to get into the tumor. Think of a solid tumor, like a dense physical structure. T-cells have a hard time infiltrating efficiently. And even if they do manage to push their way in, the local environment, the tumor microenvironment, or TME, it's incredibly hostile. It's immunosuppressive. It basically just exhausts the T-cells.
So it's like a fortress, chemically and physically designed to keep attackers out.
That's a great way to put it. And then there's the third challenge, which is a huge practical one, cost.
Yeah.
The way CAR T-cells are made now, it's highly personalized, very complex, and just phenomenally expensive. Hundreds of thousands of dollars per patient. It limits access dramatically.
Right. So we need something that can break into that fortress, fight safely once inside, and ideally be way more affordable. And the research we looked at points very strongly towards one potential solution, CAR-macrophages.
CAR-Ms.
Exactly. Arming the body's natural cleanup crew, essentially.
It sounds almost too perfect. Why macrophages?
Well, what's so compelling is that macrophages naturally have the traits that CAR T cells lack. They're designed to migrate, to get into dense tissues easily. That's their job.
Okay.
They're also professional phagocytes. They literally eat other cells, debris, pathogens. And critically, they're fantastic antigen-presenting cells. That's key for building long-term immunity.
Okay, let's dive into that. Mechanism and safety. This is really interesting because activated macrophages, they produce inflammatory signals, right? Like IL-6, IL-1, the same stuff involved in that dangerous CAR-T-CRS.
That's right.
So why wouldn't arming macrophages just lead to the same kind of severe toxicity? What makes CAR-M safer, potentially? That's probably the most crucial question. And the answer seems to lie in how they kill the tumor cell. It's a different process.
Okay.
CAR T cells and also CAR NK cells, they kill mainly through what's called CAR-dependent cytotoxicity. Think of it like setting off a bomb. They use proteins, perforin and granzyme, to punch holes in the target cell.
Right.
This causes the cell to rupture really quickly, releasing all sorts of inflammatory stuff. It's that aggressive, immediate destruction that seems to trigger the severe CRS.
So aggressive kill, aggressive inflammatory reaction.
Logical, exactly. CAR-macrophages, though, they primarily kill through CAR-dependent phagocytosis.
Phagocytosis, eating.
Yes, they bind to the tumor cell via the CRR, and then they engulf it, digest it internally.
Right.
So much more controlled, cleaner removal process, more like apoptosis, programmed cell death.
Does the early clinical data actually back this up, show the safety difference?
It really does. Strikingly so. We know CAR T cells have that whole spectrum of safety issues, high-grade CRS, neurotoxicity. But the first clinical trial reported for CAR-Ms, it was an anti-HER2 CAR-M in patients with advanced solid tumors.
Okay.
The safety profile was remarkable. They only saw low-grade CRS, grade 1 and 2. And crucially, no neurotoxicity, no graft versus host disease reported in that trial.
Wow. That's a huge difference.
It is. It really suggests that the method of cell death profoundly impacts systemic safety. Clean engulfment versus explosive rupture seems to be key.
That's a fantastic way to think about it. Now, before we get too carried away, there's still the issue of target selection, right? That applies to any CAR therapy.
Absolutely.
Even if CAR-Ms are safer killer cells, if the target antigen they're aimed at is also on healthy cells, you still risk damaging normal tissues, that on-target, off-tumor problem.
Yes, that's a universal challenge for all CARs right now. The dream target is something essential only for the tumor, maybe an oncogenic driver mutation. But most of those are inside the cell, not on the surface where the CAR can easily bind. People are working on ways to target internal bits presented on the surface. But for now, yeah, choosing a target that's truly tumor-specific without hitting healthy cells is still a major hurdle.
Okay, understood. Now, let's get back to that solid tumor fortress. We said CAR-Ts struggle to get in. Why are macrophages inherently better infiltrators?
They're just built differently. They're part of our innate immune system designed to patrol everywhere, get into tissues, respond to damage signals. One key advantage they have over T cells is their ability to secrete enzymes.
Enzymes.
Yeah, enzymes that degrade the extracellular matrix, or ECM. That's the dense sort of structural scaffolding that tumors build. Macrophages can basically chew their way through it. They tunnel through the fortress walls.
Like their own little demolition crew.
Exactly.
But, okay, they tunnel in. What about the environment inside? You mentioned it's hostile, immunosuppressive, and tumors often have their own macrophages already there, right? Tumor-associated macrophages, TAMs.
That's right. And usually those TAMs are actually helping the tumor. They're what we call M2-like. They suppress immune responses, promote growth. They're part of the problem.
So how do the incoming CAR-Ms deal with these corrupted local macrophages?
This is where it gets really clever. The CAR-M therapy aims to do two things. First, the CAR-Ms we infuse are engineered or stimulated to maintain a pro-inflammatory state, an M1-like phenotype when they arrive. They arrive ready to fight.
Okay, they bring their own attitude.
Right. And second, studies in mouse models show something remarkable. These CAR-Ms can actually reprogram the existing immunosuppressive M2-like TAMs they encounter inside the tumor.
Okay, they convert the tumor's helpers into fighters?
Precisely. They seem to activate pathways like the inflammasome, boost IL-12 signaling. Basically, they release signals like TNF-alpha, IL-12, IL-1 beta that flip those local TAMs from M2 towards that pro-inflammatory M1 state.
So they're not just killing tumor cells, they're actively changing the whole neighborhood, warming up that cold TME.
Exactly, making it much more receptive to a broader immune attack.
Okay, but that initial clinical data you mentioned, while safe, only showed stable disease in a few patients. That suggests we need more. More punch, more potency, more persistence. What's being done to optimize CAR-Ms?
Yeah, optimization is key. Early versions, first-gen CAR-Ms, they didn't last long, maybe a couple of weeks. So researchers have focused on the CAR design itself.
Oh, so.
They developed second-generation CARs with a dual signal structure. This doesn't just boost the killing activity, it also strongly pushes the macrophage towards that desired M1 pro-inflammatory state when it engages the tumor antigen.
So the CAR itself helps keep the macrophage in fighting mode, resisting that suppressive environment.
Exactly. And to help them persist, to live longer in the tumor, researchers are engineering CAR-Ms to produce their own survival factors, things like CSF1 or even cytokines like IL-12.
Does that work?
It seems to. In models, this is extended CRM persistence in the tumor beyond a month, sometimes even over 60 days if they're kept properly stimulated. Gives them much more time to work.
Okay, better CARs, longer life. Now, what about teaming them up? Combination therapy. Macrophages have that famous don't eat me signal they have to overcome, right?
Ah, yes, the CD47 SIRP axis.
Yeah.
CD47 is a protein that cancer cells put all over their surface. It's like a don't eat me flag telling macrophages to back off. SIRP is the receptor on the macrophage that sees that flag.
So block that interaction.
Exactly. When they combine CAR-Ms with drugs that block the CD47 SIRP interaction, basically cutting that don't eat me signal, the results in mouse models were dramatic. Much better tumor control. You arm the macrophage with a CAR-arm and you take off its main inhibitory brake.
That makes sense. Double whammy. What about combining with existing immunotherapies like checkpoint inhibitors?
Also very promising. Combining CAR-Ms with anti-PD-1, the standard checkpoint inhibitor, led to complete tumor control in some mouse models of solid tumors. It shows CAR-Ms can work really well alongside other immune-boosting strategies.
Okay, so we've got safety, better infiltration, TME reprogramming, and ways to boost potency and persistence through design and combinations. Let's talk long-term benefit. A huge issue with targeted therapies like CARs is resistance. Tumors evolve, they might lose the target antigen.
Right, antigen escape.
How do CAR-Ms help address that? Do they offer more lasting protection?
This goes back to one of their fundamental roles.
Yeah.
Macrophages are professional antigen-presenting cells, APCs. Meaning when they eat a tumor cell, they don't just destroy it, they digest it, break it down into little pieces, antigens, and then they present those pieces on their surface. They basically show the rest of the immune system, especially T cells, what the enemy looks like.
Ah, so the CAR-M is killing the tumor and simultaneously educating the T cells about it.
Precisely. And because they're presenting many different pieces from the eaten tumor cell, not just the single target the CAR recognizes, that can stimulate endogenous T-cell responses against a whole spectrum of tumor antigens.
That's called epitope spreading, right?
Exactly. So even if the tumor tries to hide by losing the original CAR target antigen, the immune system, thanks to the CAR-arm's presentation, has now learned to recognize other parts of the tumor. It broadens the attack.
And is there evidence this leads to actual immune memory, long-term protection?
Yes, the preclinical evidence is strong. In those mouse studies where they combine CAR-RMs with, say, anti-CD47,
Yeah.
mice that were cured of their tumors were later rechallenged with the same tumor cells, but without any more treatment, and they rejected the tumor.
Wow.
It strongly suggests that the initial CAR-M therapy induced a lasting immunological memory, likely driven by those educated CD8 plus T cells. This shifts the whole paradigm from just a direct killing therapy to something that could potentially re-educate the patient's own immune system for long-term defense.
Okay, this is incredibly promising. Let's loop back to that third big hurdle, costs and accessibility. Personalized ex vivo CAR-T is just too complex, too expensive for widespread use. Is there a path to make CAR-M therapy more like a drug?
That's the ultimate goal for many in the field. Moving from ex vivo manipulation, taking cells out, engineering them, growing them, putting them back, to in vivo gene editing.
Doing the engineering right inside the patient.
Exactly. Leveraging the macrophage's natural tendency to gobble things up. The idea is, can we deliver the CIR gene directly to the macrophages in situ, bypassing all that costly external manufacturing?
Turning cell therapy into potentially an off-the-shelf drug-like treatment, how are people trying to do that?
Nanotechnology is playing a huge role here. One approach uses lipid nanoparticles, LNPs, to deliver CRG mRNA, very similar tech to the COVID vaccines.
Okay.
In models, this led to CAR expression at a fraction, maybe 5 to 15%, of circulating monocytes, their precursors to macrophages. And even that transient expression gave significant tumor control.
Interesting, but mRNA is temporary? Any more permanent in vivo approaches?
Well, maybe not permanent yet, but more sustained or localized. Another strategy used a nanoquarter hydrogel system. They loaded it with plasmid's DNA carrying the CR gene and placed this gel right into the brain cavity after removing a glioblastoma tumor in mice.
Directly at the source.
Right. And they found about 12% of the macrophages in that area started expressing the CAR. When they combined that with blocking the CD47 don't eat me signal, it controlled tumor relapse in over 80% of the mice.
That's impressive. Suggests maybe localized delivery could be really powerful for certain cancers.
It does. It points towards transforming this whole field.
So wrapping this up, it feels like CAR-Ms really hit a sweet spot. They potentially address all three major CAR-T limitations: better safety through phagocytosis, natural infiltration, and reprogramming of solid tumors, and now this exciting prospect of in vivo delivery, making it potentially cost-effective, almost drug-like.
That's the hope, absolutely. The next steps are clear. Continue refining those dual-signal CAR designs, maybe engineer CAR-RMs, to conditionally release specific cytokines in the tumor, like type I interferon, IL-12, IL-15, to really overcome that immunosuppression. And crucially, push hard on developing those in vivo delivery systems, maybe even using LNPs to deliver everything needed for permanent gene editing, like Cas9, guide RNA, and the SAR DNA, all targeted to macrophages for site-specific integration.
Which leaves us with a really fascinating question for you, our listeners, to think about. If we get really good at the safely, efficiently editing macrophages in vivo, essentially reprogramming the body's main immune scavengers and signalers with a simple injection, what comes next?
Yeah, beyond cancer. Think about other chronic diseases where macrophage dysfunction is central, certain autoimmune conditions, maybe even some complex neurodegenerative diseases. Could they be targets for this kind of revolutionary in vivo macrophage reprogramming? It really opens up a whole new therapeutic landscape.